Since the first attempts to explain the structure and function of the human eye, each new decade has brought better understanding of it. With development of the compound microscope, the science of modern embryology was born, and with it the realization that solution of many of the problems found in the adult eye depend upon thorough knowledge of the processes of development which produced it. No part of the body just happens. Before any structure can be complete and functional, there must be a long series of interacting developmental events. Two types of activity are concurrently involved in this development. One is growth, which depends on the multiplication of cells and the subsequent growth of the new cells. The other is the directional change in shape, structure, and function of those cells. Both activities are controlled by two forces. Gene determination decides the direction in which a change can occur, while the reciprocal demands of the individual cells or parts determine how far that direction must be followed. Fundamentally, the process consists of these two activities. Change in structure and shape due to relatively different rates of growth, and change in structure and function due to differentiation and functional specialization. The first major activity in the life of the embryo is the formation of a nearly flat plate consisting of three germinal layers: ectoderm, mesoderm, and endoderm. In the development of the eye, only the first two are of importance. The neural ectoderm will contribute cells for the formation of the retina, the iris muscle and epithelium, the ciliary epithelium, the optic nerve fibers, and glia. The surface ectoderm will contribute the epithelium of the cornea, lids, glands, and the lens. The mesoderm will contribute ciliary, orbicularis, then extraocular muscles, the connective tissue of the cornea, iris, sclera, lids and orbit, and the vascular system. The next step can be seen more clearly if we watch it both from above and in section. The neural ectoderm on either side of the midline grows much faster than the other layers, forming two longitudinal folds. These folds begin differentiation of the central nervous system. By the 20th day, the mesoderm has formed several pairs of primitive segments, or somites, which are visible as small bumps on the surface. Possibly due to the more rapid growth of the exterior surfaces, the folds begin to lean toward each other. As their edges touch, they fuse, and the groove between them becomes a tube. From now on, our interest will center on the anterior end of the embryo. Let's look at a section cut through here. The two mounds are the neural folds, which haven't yet closed. The surfaces facing each other are neural ectoderm. The outer surfaces are skin. The little depressions, which now appear, are the optic pits, the first signs of the developing eye. Here they are in a model of a 10-somite embryo. The neural tube has closed anteriorly as far as the future midbrain. As the neural folds close, the pits push out, away from the center of the nervous structure and against the surface ectoderm. The embryo now has 20 pairs of somites, and the anterior end of the neural tube is complete. The bulge on each side of the head is caused by the outward extrusion of the growing optic pits, which are becoming sac-shaped vesicles. As the overall development continues to 25 pairs of somites, the area between vesicle and brain constricts to form the optic stalk. So far, there has been no differentiation in the neural ectoderm forming the vesicle. Now, however, the vesicle stage has reached its peak, and its form will change radically in the next few days. The process of differentiation will begin, leading to the formation of pars optica retinae from the anterior part and pigment epithelium from the sides. The region between the two will form the epithelium of the iris and ciliary body. The forward part of the vesicle wall grows thicker and flatter, and a similar change occurs in the surface ectoderm adjacent to it. This will become the lens. In the photomicrograph, this disc-shaped thickening over the area between the lips of the future optic cup is the first manifestation of lens induction by the optic vesicle. It is the site of the earliest specific lens proteins. In order to facilitate identification of parts, the same arbitrary colors used in the models and drawings will be substituted in some of the photomicrographs for the colors produced by usual stains. The nuclei in the retinal area have begun to move toward the inner edge, the first differentiation of the retinal cells into layers. These areas are filled with mesoderm. This part of the vesicle and the corresponding lateral portions begin to grow more rapidly than the other part. Again, this difference in rate of growth results in buckling. Both the optic vesicle and the lens placode begin to fold inward. Invagination of the lens placode is accomplished by a synergistic elongation of its cells with a contraction of their apical cytoplasm and terminal bar system. 2 days have elapsed since the previous photomicrograph. The optic vesicle is becoming a cup with a little space between it and the lens pit. This space is the future vitreous cavity. And besides filamentous strands of neural and surface ectoderm origin, it contains a few mesodermal cells. The loose cells in the lens pit are presumably discarded. In a model of the same embryo, we see that the cupped shape of the optic vesicle is imperfect because of a so-called choroidal or fetal fissure grooving the vesicle and its stalk on the lower surface, nearly to the brain. A day later, the lips of the lens pit have closed to form a complete vesicle, which is still attached to the surface ectoderm. As the fundus of the optic cup continues to retreat inward, the space between it and the lens vesicle increases. It is being filled primarily by non-cellular material, although blood vessels and some mesodermal cells are present in the early stages. The edges of the fetal fissure have been growing toward each other and now begin to fuse. This model shows the appearance of the fissure at this stage very clearly. The stalk has narrowed considerably in the last several days. Let us look at some of the details given by a photomicrograph at the same age. This is where the fetal fissure has already closed. The space between the retinal and pigment epithelial layers almost always appears greater than it really is because of tissue shrinkage during preparation. In animation, we'll take this into consideration and won't try to match the photomicrograph exactly. It takes about 3 days for the fissure to close completely. All that remains of the fissure is a small notch in the edge of the cup and an opening in the stalk just behind the eye. The stalk itself is quite slender, joining the cup at a more central point than heretofore. From this point on, the general shape of the eye doesn't change much. By superimposing an adult eye, reduced to the same size as a 36-day-old cup, we see that most future changes are localized ones resulting from differentiation toward specialized function. Changes of this type are extremely complicated, and it will simplify matters greatly to divide the eye into regional sections, following each one through to term. The lens vesicle has just separated from the surface ectoderm. It consists of a single layer of cells covered by a basal lamina, which through appositional growth to its epithelial surface will acquire more layers and become the lens capsule. The cells in the hollow of the lens pit were trapped inside the closing vesicle. The anterior wall of the vesicle is a layer of cuboidal cells which remain undifferentiated except at the equator. The columnar cells of the posterior portion are steadily growing longer. They are the primary lens fibers. Specific lens proteins or crystallins are synthesized within them. In the week since the vesicle broke away from the surface ectoderm, primary fibers have completely obliterated the lumen. For another week, as the central nuclei fade and disappear, growth of the lens is simply a matter of growth of the primary fibers, and a roughly spherical shape is maintained. Then a second stage in the lens development begins. In the region of the equator, some of the cuboidal cells elongate to form the first secondary fibers. They are thick at the equator, but taper as they grow toward the front and back of the lens. Their tips extend around the primary fibers and beneath the layered capsule. Their ends do not meet those of other fibers at a single point, but on planes organized essentially vertically to the surface, and linear sutures result. Because of this arrangement, no fiber ever becomes long enough to reach completely from pole to pole. The posterior suture is formed when the posterior central cells lose their nuclei, become separated from their basal lamina, and migrate inward. Curved lens fibers result, with the superficial ones being the longest. The lens is becoming less spherical. Its shape and its orientation with respect to the optic axis continually adjust to the developing eye. This is partly regulated by the neural retina and the peripheral mesenchyme. A week later, the equatorial diameter averages 17% greater than the antero-posterior. And less than a month after that, the average difference has increased to 27%. Maturation involves a progressive general structural densification. Fibrillar material is increased within the cytoplasm, and cell organelles are decreased. The successive parallel layers of interdigitating elongated lens fibers become very tightly opposed, and the inner nuclei disintegrate. The pattern of development in the lens is now pretty well set. Succeeding generations of cells extend from the equator anteriorly and posteriorly beneath the capsule. The anterior suture line is Y-shaped, which is inverted at the posterior suture. A section through the lens of a 5-month fetus represents the inner layers of the adult lens seen with slit lamp. At birth, both sutures lie beneath the capsule, the surface enclosing beneath it the fetal nucleus. Development continues at a gradually diminishing rate through gestation and even into adulthood. Our second detailed section involves development of the retina, the optic nerve, and the vitreous. On the 35th day, there are a few pigment granules in the cytoplasm of the pigment epithelial cells. The vitreous cavity is filled with blood vessels, filamentous strands, mesodermal cells, fibers, and secretions from the lens and retina. Nearly all nuclei in the retina have migrated toward the outer edge. Between the nuclei and the internal edge, the long, slender fibers of Müller have begun to appear. At the internal surface, they have spread out and joined, forming an internal limiting membrane. A few retinal nuclei begin to migrate back toward the internal limiting membrane. This migration, which begins development of the inner neuroblastic layer, starts in the region of the future fovea and spreads very slowly toward the periphery. The beginnings of a definite second nucleated layer can now be seen, separated from the first by the transient fiber layer of chivets. Formation of the inner layer continues as axons from ganglion cells reach out to the internal limiting membrane and extend along it toward the stalk. They will form the optic nerve. Whereas the eye itself developed out away from the central nervous system, the optic nerve grows in the opposite direction, back to the brain. Primary vitreous is composed of filamentous substance of neural and surface ectodermal origin from the lens to which vascularized mesenchymal strands had joined through the choroidal fissure. The origin of the secondary vitreous is controversial. The main contribution is believed to come from the vitreous fibers of retinal and Mullerian footplate origin. It surrounds the primary vitreous, and its fibrils are firmly attached to the basement membrane covering the retina. The granules in the pigment epithelial layer increase steadily, and the epithelial cells are being separated from the mesoderm by the formation of Bruch's basement membrane. In the retina, the external limiting membrane, consisting of a series of intermediate junctions between the apical ends of the Mullerian cells and the future photoreceptors, has appeared. The first nerve fibers have reached the optic stalk and are growing into it. At the junction of the stalk and retina, the fibers pass under some of the cells which surround the hyaloid artery. These cells will grow into a cone-shaped mass of glia known as Bergmeister's papilla. As the fibers extend, growing between the cells, the nerve grows in thickness. Many inner cells of the stalk vacuolate and are replaced by axons. Others become converted into neuroglia. The outer cells are also differentiating into the stellate glia cells that will support and nourish the nerve. Here we see how the optic stalk looked just before the fibers had entered it. And 2 days later. In another week, the entire optic stalk is filled with nerve fibers enveloped by a pile connective tissue capsule. In the retina, the inner neuroblastic layer has progressed over half the distance to the rim of the cup. The still undifferentiated ciliated cells of the outer layer, which will eventually become rods and cones, are in close apposition to the pigment epithelium. 3 weeks later, the external limiting membrane is well established by the Mullerian fibers, and a few cone cells are protruding near the posterior pole of the retina. A narrow outer plexiform layer and the first synaptic lamellae and vesicles in the cone pedicles are beginning to form. The inner neuroblastic layer has split, and the fiber layer of chivets will soon disappear. The ganglion cells are forming a definitive layer. A narrow inner plexiform layer is appearing and contains a few synapses between processes of the inner nuclear and ganglion cell layers. 10 days later, there are cones throughout the retina, but rods are still undifferentiated. The cells of Bergmeister's papilla proliferate to form a glial sheath around the hyaloid artery. Both sheath and papilla atrophy with the hyaloid vessel system before birth, the amount of atrophy determining the depth of the physiologic cup in the center of the disc. The future macular region is indicated during the 6th month by an accumulation of ganglion cells, local increase of cones to the exclusion of rods, and thinning of the plexiform layers. During the next month, there is a gradual spreading out of the ganglion cells from the central foveal depression. At birth, the foveal ganglion cells are reduced to a single row. Outside the foveal region, the rods are now visible. The optic nerve has been in contact with the brain for 5 months. The lipoid glial cell membranes surround the nerve axons, forming a thin myelin sheath. This medullation spreads slowly toward the eye, but is not completed until the 9th month. All layers of the retina have now differentiated except at the fovea. With the completion of the medullation process, the retina is ready to function. We again return to day 36, this time concentrating our attention on the anterior regions and the orbit. Early in the 6th week, the mesenchyme near the front edge of the cup begins to condense as the first step toward formation of the sclera. The mesoderm, separating the surface ectoderm from the lens vesicle, forms a layer of cells which will become the posterior corneal surface, descamase mesothelium. Additional mesodermal cells apparently press between the epithelial and endothelial layers from the periphery to form the corneal stroma. Similarly, nebulous strands of mesoderm grow inward between the lens and the cornea, forming the pupillary membrane and delimiting the anterior and posterior chambers. The anterior chamber, at first small in diameter, will grow peripherally. The cornea is continuous with the newly formed sclera, which now extends as far back as the equator. These condensations, which appeared 10 days ago, will form the extrinsic ocular muscles. In the cornea, collagen synthesis by the keratocytes of the stroma is underway. This is surface epithelium. And here we see descamase mesothelium, or the endothelium. Bowman's and descamase membranes have not appeared. The scleral condensation has now reached the posterior pole of the eye. Between the lens and the margin of the optic cup, the vitreous fibers line up to form the marginal bundle of Drouot. The rim of the cup is growing forward under the pupillary membrane and will soon begin the epithelial part of the iris. Just back of the rim, the 2 layers begin to form the ciliary body. Folds appear where the ciliary zone expands more rapidly than the overlying sclera, and these will form the ciliary processes. 10 days later, both layers are growing together, except at the tip. The long ciliary muscles develop from the mesoderm between the ciliary body and the sclera. This is a posterior view of the ciliary processes with the lens removed. They remain a constant 70 to 75 in number. The surface is covered with fine filaments instead of a limiting membrane. The canal of Schlemm, which may be seen about this time, is a ring-shaped vessel running completely around the eye. Through it, the anterior chamber will be drained into the venous system. The tiny protoplasmic processes sprouting from the ciliary body are zonula fibers. They grow right through Drouot's bundle, which almost immediately begins to atrophy. As the zonula fibers attach to the lens capsule and thicken, they become the suspensory ligament for the lens. In the cornea, collagen synthesis has proceeded rapidly. Beneath the epithelium, the acellular collagenous Bowman's membrane is just appearing. The vitreous has developed to an essentially adult structure. The cornea, because of its own growth and that of the anterior chamber, has assumed a curvature, which, for the first time, differs from that of the eye as a whole. Traces of the sphincter can be seen. The iris stroma is growing in thickness, and the cells of its posterior layer are becoming pigmented. This process will continue until it reaches the root of the iris. Just behind the ciliary processes, we begin to see the pars plana. By the 6th month, the anterior chamber has opened up to the scleral spur. The sclera itself is thickened all around the globe. A network of scleral tissue has penetrated the optic nerve to form the lamina cribosa. By term, it will have nearly adult structure. Now the sphincter is invaded by connective tissue and blood vessels. By removing the pigment granules, dilator fibers are seen forming in the outer pigment epithelial layer. The pupillary membrane begins to atrophy, and fibers of the circular muscle appear in the ciliary body. The shape of the anterior chamber, especially at the angle of filtration, is nearly final. By the 8th month, the sphincter is complete and functional. By the 9th month, the ora serrata has receded to the posterior edge of the ciliary muscle, leaving a wide pars plana. The structure of the trabecular meshwork and Schlemm's canal approaches that in the mature organ, and secretion of aqueous humor has begun. Most of the pigment in the iris stroma appears after birth, the amount depending largely on the general coloring of the individual. The regions in the anterior part of the eye proper, with the exception of the dilator muscle, are now ready to function. We have watched all the major developments in the optical system of the eye. In addition, we have built a background for understanding the very intricate development of the vascular system. On the 27th day, several small vessels appear in the mesoderm around the optic vesicle, and leading from the internal carotid artery, extend a short way forward. This plexus is the primitive dorsal ophthalmic artery. The venous plexus into which it drains will become the cavernous sinus. When the branches of the artery have developed to the front of the cup, some of them turn inward and hug the fundus, while others anastomose along the rim to form the annular vessel. Where the internal carotid artery disappears behind the stalk, another vessel can be seen branching from it and leading to the far side of the cup. This is the primitive ventral ophthalmic artery. We have changed our position. When the branches of the artery have developed to the front of the cup, some of them turn inward and hug the fundus, while others anastomose along the rim to form the annular vessel. Where the internal carotid artery disappears behind the stalk, another vessel can be seen branching from it and leading to the far side of the cup. This is the primitive ventral ophthalmic artery. We have changed our position here and are now looking at the cup from below. As the choroidal fissure closes, the network of vessels inside the cup is trapped, except for one branch which still connects with the ophthalmic through a small opening in the stalk behind the cup. This now becomes the hyaloid artery. The network of small vessels at its forward extremity has formed a tunic around the lens, and in addition to its vascularizing function, it contributes to the growth of the vitreous. Veins begin to appear in the optic nerve, paralleling the hyaloid artery. The choroidal network of venous channels has differentiated in the mesoderm, adjacent to the pigment layer of the cup, and through it blood is drained to the upper and lower venous plexuses and thence into the cavernous sinus. The dorsal ophthalmic artery has made a noticeable migration back along the internal carotid, so that it now approaches the opening in the stalk from a position much lower than heretofore. The branch of this artery, which continues outside the cup toward the annular vessel, is the temporal long ciliary artery. Meanwhile, the ventral ophthalmic artery is anastomosing with the definitive ophthalmic, and its distal portion will become the nasal long ciliary. The remainder of the ventral ophthalmic will soon disappear. The ophthalmic artery is now the only branch from the internal carotid to the eye. Meanwhile, the venous system has further developed, forming the vortex and ophthalmic veins. Inside the eye, the anterior chamber has just appeared, and the pupillary membrane behind it is being fed by arterial loops or arcades sprouting from the annular ring. One can get a better idea of the developing arcade vessels by watching them from the front. At the sides of the globe, the two long posterior ciliary vessels come forward. They anastomose with the anterior vascular net near the annular vessel to form the major arterial circle. Now, arterial blood is fed into the anterior vascular capsule of the lens by both hyaloid and ciliary arteries. However, the job is taken over more and more by the ciliaries as the whole vascular system within the primary vitreous begins to atrophy and the annular vessel slowly disappears. The primary vitreous stops growing and appears relatively shrunken. Its region will be identified as Cloquet's canal in the adult. The cup continues to grow. The internal vessels do not. Circulation through the vessels appears to decrease, and the branches break loose from the hyaloid. In the choroid, the layer of larger venous channels draining directly into the vortex veins is apparent for the first time. Note that there is no pigment in the choroid layer. Toward the end of the 4th month, two arteries sprouting from the hyaloid begin vascularization of the retina. As the iris stroma grows in thickness, branches from the major arterial circle enter it. One vessel penetrated each ciliary fold, running parallel to its length. In the choroid, all the adult layers are now present. Pigment granules are noticeable by the 7th month. Returning to a front view of the eye, we see the arcade vessels at the height of their development. A number of small branches of the major arterial circle now grow into the peripheral portion of the iris stroma, and by the 7th month, several layers of vessels are distinguishable. Their relationship is more apparent in section. Most superficial are the arcades. Beneath them are the veins and arteries vascularizing the iris stroma. The pupillary membrane begins to atrophy. Its vessels shrink, and the blood flow stops. In the retina, vascularization has extended throughout. In the iris, the minor vascular circle is formed by the apices of the remaining arcades. The branch vessels leading from the major circle to the ciliary processes form a large part of the definitive circulation. By the end of the 8th month, atrophy of the hyaloid system is almost finished. The pupillary membrane has nearly disappeared. The permanent vascular system is complete. In our final detailed section, we shall look briefly at the developments in the facial region. At the same stage as the section we have seen, the maxillary process extends to the opening of the nasal pit, touching the nasal processes. The maxillary mesoderm is therefore in contact with the paraxial mesoderm surrounding the eye. The surface ectoderm covering both of these is thicker where it lies in the groove between them. The maxillary material grows up and over the thickened ectodermal line, fusing with the lateral nasal process and paraxial mesoderm, thus burying the ectodermal cells lining the groove. According to some studies, this cord of cells forms the nasolacrimal duct and sac. Others find that this cord fragments and the nasolacrimal duct forms from the portion nearest the eye, which grows down toward the nasal pit, reaching it late in gestation. According to both theories, a lumen is established shortly before birth. This growth of the maxillary process produces the lower lid, and about the same time, the upper lid fold also appears. The growing lids meet first at the outer canthus. A few buds appear on the upper and outer portion of the conjunctival sac. These will form the lacrimal gland. If we take a section through here 3 weeks later, we now see many more separate buds, showing that the gland is well on its way. The fusion of the lids takes only a few days. There is a definite orbicularis now, and the lash follicles begin to form. 2 weeks later, the meibomian gland appears. By the end of the 5th month, the meibomian glands and the eyelashes are well developed, and eyelid adhesions begin to break down. The eyelids are now complete. Although it was necessary to observe development after the formation of the optic cup in piecemeal fashion because of the great amount of detail, it is also necessary to gain an overall picture of the ways in which the details are related to each other. By the end of the 1st month, the optic vesicle is deeply indented and is grooved by the choroidal fissure. The cup is about 3/4 of a millimeter in diameter. As compared with the 7 millimeter length of the whole embryo. The angle of the optic axes is about 160 degrees. During the 2nd month, the lens vesicle is completed and detaches from the surface ectoderm. The inner and outer neuroblastic layers of the retina are formed. The choroidal fissure closes, trapping the hyaloid within the cup. The primary vitreous is complete, and formation of the secondary vitreous is underway, with its fibrils in intimate contact with the basement membrane covering the retina. The optic nerve fibers begin growth through the stalk, and toward the front of the cup, scleral condensation develops. The vascular tunic of the lens forms, and outside the globe, the long ciliary arteries appear. Primary lens fibers fill the lumen of the lens vesicle, and a capsule surrounds it. The anterior chamber appears between the cornea and pupillary membrane. Secondary lens fibers and the first suture are formed. The angle of the optic axes has been reduced to about 80 degrees. During the 3rd month, the intraocular vascular system reaches its peak and begins to wane. The lids have met and are fusing. The edges of the cup begin to form the ciliary body, and the long ciliary muscles appear. Drouot's bundle is formed and then regresses, leaving the growing zonula fibers in this region. The ciliary processes and the epithelial part of the iris have developed. The cone nuclei differentiate in the retina, and a narrow outer plexiform layer and the first synaptic lamellae in cone cells are noted. Synapse formation is also commencing in the inner plexiform layer. The canal of Schlemm appears, and the angle of the anterior chamber is just even with it. Both the choroid and the sclera are quite well formed, even in the posterior hemisphere. The lens achieves its basic, somewhat flattened structure. The angle of the optic axes is now 60 degrees, approaching the adult condition. By the end of the 4th month, vascularization of the retina has begun. The vitreous has reached adult appearance, and the cornea has developed a curvature different from that of the eye as a whole. The diameter of the eye is about 5.5 millimeters compared to the 110 millimeter length of the fetus. About the middle of the 5th month, the anterior choroidal vessels enter the ciliary processes, and the anterior chamber extends to the scleral spur. By the end of the month, the structure of the lids is complete, and their adhesions break down. The major arterial circle supplies most of the iris and ciliary processes. The dilator fibers begin to form in the pigment epithelial layer of the iris. The vascular arcades of the iris are at their height, and medullation of the optic nerve begins near the brain. Rods and their synapses noticeable during the 5th month are, during the 7th month, differentiated throughout the retina, so that except for the macula, it is similar to the adult and is well vascularized. Vascularization is also complete in the iris, although the pupillary membrane is still present, and in the ciliary body, the circular muscle fibers are developing. The appearance of the trabecular meshwork and of the anterior chamber, especially in the filtration angle, resembles that of the adult eye. All layers are now present in the retina, but the fovea centralis is incomplete. In the 8th month, the sphincter becomes complete and functional. The hyaloid vascular system has disappeared, except for slight traces of the posterior lens net. By the end of the month, the eye has grown to somewhat over 13 millimeters, and the fetus to a length of nearly 300 millimeters. In the final month, the minor vascular circle of the iris is present, and we find the arteriolar branches from the major circle to the ciliary processes, forming a large, well-defined circulatory system. Medullation of the optic nerve reaches the retina, completing the neural pathway. At term, with relatively few exceptions, the eye is a complete sensory organ, ready to function when the first rays of light pass through its pupil.